I want to start off by thanking the organizers for including me in the program. As Cecil said, I'm relatively new to cultivated meat. I've only been working on it for roughly around 18 months, so this is pretty exciting for me. So today I'm going to talk about rational design of cell culture media using metabolic models. As Mark said yesterday, the growth media is still a significant cost or portion of the cost for cultivated meat, right? It's around 30%. And if we look further into how that is broken up among the growth media components, again, as Mark said, the amino acids are the significant cost here. So the growth factors are only a very small portion because you use so little of them. So one thing that we can do in order to optimize the amino acid ratios is to use metabolic modeling. And metabolic modeling allows you to perform numerous simulations outside the lab in a fraction of the time it takes to do this in an experiment. And so it can really save you time and money. So I'm a chemical engineer by training, and what chemical engineers typically do is design chemical plants. And in order to design these chemical plants, we have these complex networks describing the different unit processes that are needed to produce the chemical. So in much the same way, we look at a cell and we build a metabolic model. So we're quantifying all the, or enumerating all the metabolic pathways that are in the cell and then modeling them so that we can try to have the cell make what we want. So one of my favorite analogies for metabolic modeling is a traffic map. So if you're looking at, to get from point A to point B, you Google the map, right, and it will tell you how to get there. When we're looking at metabolic modeling for media optimization, instead of looking specifically at how the traffic pattern is, right, it's more thinking of a tollway. So we really only care about how the cars come in at the beginning and leave at the end. And so for this case, for example, I put this in reverse order, sorry. If we're thinking about driving from Cologne to Maastricht, right, and we're pretending it's a tollway, we really only care about how we get there, or how many cars are leaving Cologne and how many are arriving in Maastricht. And much the same way, we really only care about how much glucose is going in and how much lactate is coming out. But we can also look further to see exactly how these things occurred. So here's the traffic map analogy. Sorry, I reversed these things. So if we think of the cell that we're modeling as Europe, and the specific metabolic pathway that we're interested in is the route from Cologne to Maastricht, right? You'll notice that there's a backup there in Aachen, and my husband's favorite team is in Aachen. So I'm going to say that Alemannia Aachen got promoted from the 3rd division to the 1st, and that's why there's a backup. Everyone's going to the game. So what can you do to get around it? You could expand existing roads. So if we're engineering the cell, that would be overexpressing enzymes in that specific pathway. You can redirect traffic, so you could tell everyone to get on the train instead, right? In metabolic engineering, that would be finding an alternate pathway to overexpress the enzymes in. And then finally, you could build a new road. So you could find a pathway in a different organism and put that in. So that's how we use the metabolic maps. How do we build them? The first thing we have to do is perform a metabolic network reconstruction. And so in order to do this, what we need is the genome sequence of the organism that you're interested in creating a metabolic network for. In my lab, we've built an algorithm so that we can leverage the work that's already gone into other metabolic models. And so what we're looking at is we combine published metabolic scale models, published genome scale metabolic models, with the organism that we're interested in. We can do a blast search to see if the proteins are similar, and then it creates an automatic first draft metabolic network. So this takes about 20 minutes. When I was a graduate student doing this, it took me all 5 years. So this is a great advance, but it still requires a lot of manual curation, and we'll talk about that in a little bit. So again, once we have the metabolic network reconstruction, the next thing we have to do is to write a mass balance. So again, this is a very normal chemical engineering thing. We're always worried about mass and energy balances. So if we have the metabolic network reconstruction, and we're going to do it on metabolite E there, what we can say is the accumulation equals what's coming in minus what's going out plus generation. So for this particular metabolite, the flux in is V10 and V13, and the flux out is V11. So we get the change in the concentration. This is the only math you'll have to listen to for the whole talk. We get the change in the concentration of V with respect to time equals the fluxes in minus out. And then this last term is the growth rate times the concentration, and we usually ignore that because it's several orders of magnitude smaller than the fluxes themselves. That makes it nice because then it's a linear equation. We throw that into matrix form, and we can solve for the fluxes themselves. And so we get, this is a vector of the changes in concentration into the cell, the stoichiometric matrix, which contains all the information of all the reactions in the cell, the stoichiometry of all the reactions. And then this vector is what we're solving for. Typically, if you're doing this on a genome scale, you have thousands and thousands of reactions and not enough experimental data to solve it directly, so what we have to do is add constraints. The first thing is steady state, so we're assuming that nothing is accumulating within the cell. We define substrates as being taken up by the cell, products being excreted, and then we also add experimental data. And then lastly, it's an underdetermined system, so you have to solve it using an objective function. For bacteria, that's usually maximized biomass. For other organisms, it can be, maximizing ATP or minimizing ATP or minimizing byproducts. It really depends on what you're trying to investigate. So we've done this for porcine cells with funding from the Cultivated Meat Modeling Consortia. I have 3 wonderful students working on this. Oops, sorry. So Sandra and Victoria are here, so if you want to ask them more about it, you can find them at the conference. And Mark he didn't get to come. But basically what we've done is we've taken the genome of pigs, we've run it through our algorithm, we got a first draft network. We did quite a bit of manual curation, so that means making sure that the stoichiometry of the reactions are the same, making sure that there's no gaps, removing duplicates and things like that. And then a big part of this is the experimental data. So the experimental data really determines how accurate your metabolic prediction is. So we'll talk about that in a little bit. And then what we can do is run it through the simulation and identify, the components that are the most important for media or optimized ratios and things like that. So here's the current state of our metabolic network reconstruction. So it includes around 12,000 metabolic reactions in different compartments. There's a lot of exchange reactions because there's a lot of compartments in the model, and about 8,500 metabolites. So the key experimental data that we need in order to make this model work, the first is biomass composition. And from previous work in algae, we know that it's very highly dependent on the growth conditions. So you see here the biomass changes in a single algal species depending on what the substrate is. And so in order to maximize our accuracy of our model, what we did was we worked with the Colorado State University Department of Animal Sciences to get tissue samples from live animals so that we could most closely match the biomass composition that we're trying to achieve with cultivated meat. So we have adipose tissue, muscle tissue, and connective tissue. And of course, these aren't pure samples, right, because they come from the animal, but they are meant to represent at least a highly concentrated version of each different cell type. And then aside from the macromolecules, we also have to know the relative amino acid composition and the fatty acid profile in order to model the biomass accurately. So you can see here we did this for the 3 different types of tissue, and they have pretty significant differences in biomass composition. And because biomass is what you're optimizing, if your biomass formation equation is wrong, then you're going to have incorrect predictions from your model. And then we also needed to have experimental constraints. So these are from a cell line, a muscle satellite cell line, and we're just measuring the uptake or the excretion of different components in the media. So we have glucose, of course, is their substrate, the different amino acids, and then we're measuring the production of lactate and ammonia. So once we combine all of that into the model, we can run simulations. The first one we did was to minimize byproducts. So we wanted to minimize the production of ammonia and lactate. And so we see some general trends. If you increase the uptake of histidine, valine, and glutamine, for both byproducts, you'll reduce the production of ammonia and lactate. If you decrease lysine, you'll also reduce the ammonia and lactic acid production, but that one's a bit tricky because lysine has a pretty big play on biomass productivity, right? So you have to weigh the relative weight of how much you want to reduce your byproducts versus your biomass. And then what we did was look at amino acid supplementation. So we ran simulations to see which amino acids would have the greatest impact on growth rate. So what we saw was the addition of tyrosine, methionine, phenylalanine, leucine, and lysine all had simulated in the gray bars the highest impact on growth. So we ran these in an experiment, and what we did see was that phenylalanine and leucine and lysine had a pretty big impact on growth. And then we went back to the literature, and what we found was that agriculture, which has been raising these animals for a very long time, right? They already knew this. So for whole animals, they've already identified that lysine, leucine, and phenylalanine and tyrosine are the most important amino acids to feed pigs. So one thing is we don't have to reinvent the wheel with cultivated meat, right? Where there's already years and years of nutrition data from agriculture that we can rely on. But you do notice that these growth rate predictions are wildly inaccurate, right? Those would be like far faster than a cancer cell line. So what can we do to improve the accuracy of these models? So we can add improvements. So this modeling method is called flux balance analysis. It's very simple. It has relatively few, it's very easy to implement, and it has relatively few barriers to entry. I mean, all you need is the genome sequence. So we can add additional complexity to the model in order to improve accuracy. So some examples of which are thermodynamic constraints. You can add transcript and protein abundance. So we've been working with Panome Bio. We have a beautiful data set on protein abundance that we're going to integrate into this model in the future, and as well as metabolomics. You can add dynamic modeling, so you can model not just a steady state, but cells through a differentiation process or through a batch process, spatial modeling, or signaling, which would be important because then we get the impact of the growth factors on the metabolism itself. And so I haven't had the opportunity to implement these on a cultivated meat strain yet, but we have done this in algae in my lab. So I'm gonna talk to you about how we've improved the simple FBA to model more complex things. So my lab came up with a framework called multi-scale, multi-objective systems analysis, or MMOSA. And what we're doing is we're taking this very simple metabolic model and adding additional complexity so that we can improve the accuracy for heterogeneous systems as well as more dynamic processes. So cells don't like to just optimize one thing. They like to optimize multiple things, so we're using multi-objective. So instead of just optimizing biomass, we can optimize biomass while minimizing lactic acid or something like that. We can take into account the differences in the vessel for mixing and nutrient diffusion. For algae, the circadian clock is very important, but you can imagine that any type of changes in gene expression, for example, during differentiation, can be accounted for. The biomass changes in algae from day to night, so we account for that. And then we also have the interactions between cells. So we did this for a model algal system, and we're basing it mainly on transcriptomic data. So we have transcriptomic data of cells grown in a 24-hour day-night cycle of 12-day, 12-night. And so we have data on all 17,000 genes and how they change over the course of this one day. For those of you who aren't familiar with plants, rubisco is the carbon-fixing enzyme, so this is just an example of the gene expression data. In order to model it in finer time points than the 2-hour resolution that we have to convert this to continuous. So what we do is we identify a handful of potential functions that can describe that data, and then we weigh each one using Isccm's information criterion, which basically just tells us how much information we lose by choosing one over the other. And then we can get a fit to the data for each gene. So once we have this, we can implement it as a constraint on the model. So if the gene is available, then the metabolic reaction associated with it is also able to function, and vice versa. And so what we see here is this is a metabolic map of an algal cell. It's a very dynamic process. It's going from day to night. And so if I click on it, then you see how the fluxes change. So the thickness of the arrow tells you how much carbon is going through that reaction. And so right now we're in nighttime, and now we're switching to daytime, so you'll see more fluxes in the plastid there. So this was the first example of being able to use flux balance analysis to describe a dynamic process, such as growth in diurnal conditions. Another cool thing about the model is that we did a knockout analysis, so we looked at knocking out every single gene in the model and the impact on growth. And for algal systems, we're looking at biofuels, so we want cells that grow fast and have a high lipid content. And so what we saw is that there's a handful of mutants over here that have increased biomass and increased lipid. So what we did was we identified some of them. One that made logical sense was ATO1, which is a knockout of the beta oxidation pathway. So if you block the consumption of biomass or of fatty acids, then the cell accumulates fatty acids, right? We found that mutant in a library. We grew it, and indeed, we were able to show that these mutants grow faster than the parent strain. And then when we look at the lipid profile, you can see that they also accumulate more lipid. And again, this was the first time that we've shown the ability to predict changes in day-night growth that actually happened when we did the experiment, because typically these models are only used for steady state, so that would be continuous light or continuous dark. And then something else that we're working on is that we can track the cells in space and time using this framework. So on the left there, this is nutrient tracking. As it gets darker red around the cells, you can see that they're taking up CO2. So you can imagine we can use this to model the cells in the reactor and account for the heterogeneous conditions, because nothing is ideally mixed, right? And then the other one is showing self-shading. So if we're lighting this column from the top, cells at the top are absorbing light and blocking it from the cells below. So this is just another example of how we can use this framework to track individual cells in space and time. Okay, so I hope I've shown you that even the simplest version of flux balance analysis can help you reduce the R&D time to optimize your cell media. You can do these simulations in minutes instead of weeks in the lab. And then if we add more complexity to these models, we can use it to model dynamic processes and minimize the impact of heterogeneity on productivity. So with that, I'd like to thank our funders. So the Cultivated Meat Modeling Consortia funded this project. We've been working with Panome Bio on proteomics and metabolomics data. And then Sandra and Mark and Victoria are the ones that really did all this work. So thank you, and I'm happy to take questions.
Thank you so much. Great talk. Do we have any questions? Yes, we have. Andrea. Thank you very much for the very interesting talk. I'm curious if you could speak more to the multi-objective function selection, and specifically, how do you actually integrate those objective functions, and then how do you choose which ones you like to integrate? And then lastly, are there any downsides to integrating more and more objective functions?
I'm going to take a stab at the last one. So yes, it costs computational time the more complex you make the objective function, right? But computational time, at least these simulations are fairly easy so far. So it's kind of negligible. When you get into the more complex modeling, like I showed for the algae, then that does have an impact because those simulations take maybe a week to model a day. How we determine which objective function really depends on what matches the data. So I can't interview the cell and say, like, what is your objective, right? So we have to just guess and check and see which one matches the data the best. And as far as multi-objective, how we implement it, so I showed the Pareto front. Right, so this is, this is a typical Pareto front. So that's just the optimal solution if you have two objectives, right? So we try to actually calculate that a priori so that we can minimize computational time. But yeah, implementing it, it's just like a line of code in the model of do this and this at the same time. I hope I answered your question.
Yeah, thanks.
Any more questions? Yes, in the front there. Here, in the front.
Beautiful work. There is a lot of connection between metabolism and epigenetics. All of these metabolites can then be used by enzymes in the nucleus. Have you thought about integrating these models to also look at what would be a favorable media for epigenetic remodeling during cell differentiation to favor certain particular histomarks and whatnot?
I have not, but that is something that we're looking at in the future.
So there was a question in the back, on the left.
Thank you. This is a super interesting talk. I was just wondering what your strategy was for reaction gap filling when you're building your pig gem, and then what kind of tests are you doing to ensure that your model is functioning as expected by like experimental conditions?
Yeah, so the gap filling, well, we use MeMote, which is like a common scoring of how good your model is. And then that algorithm that I showed from our lab does a lot of the gap filling automatically because it will look at other published models. And the advantage of that is that we're leveraging the work that's already been done because manual curation is an extensive effort, but it still requires manual curation on top of that. And then how we're checking to see that it fits experimental data, it's a lot of running the simulation and I guess rule of thumb. I've done this for 20 so years, so I can get a feeling of whether or not the predictions are actually something physiological. So it's a lot of running the model, looking at what comes out, changing the constraints, and then doing it again. Unfortunately, that has not been automated yet because maybe we need to do that with AI or something, but.
Thank you.
Yeah.
Any more questions? Yes, there is one in the middle back there. It's a lot of running for the young.
Very cool to see metabolic modeling in the cultivated meat space. So maybe a little bit of a cynical take because I spent a lot of my PhD also doing metabolic modeling for mammalian cells. But you mentioned that the sort of underdetermined space due to the size of the models and the constraints that you put on means that you need to make some assumptions and add on additional constraints and so on. Even so, you still end up with a very, very underdetermined system, right? You're maybe measuring 20 amino acids and your model is 12,000 reactions, right? So how do you sort of reconcile the difference between these models telling you an optimal state and the cell maybe having a mind of its own and doing what it wants? How can you sort of extract useful information from the models when the solution space is so large and difficult to explore experimentally?
It's a great question. So, of course, one thing that I didn't mention is biology really loves redundancy, right? So they're never going to perform in an optimal state. So one thing the models can help us with is determining the theoretical yield. So that's like the most optimal condition. So it's kind of a screen to see if it's even worth going after. And then another thing that my lab does that I didn't talk about today is we do experimental carbon measurements. So we can feed the cells 13C labeled substrate and track that through metabolism and get an actual picture of what's going on in the cell. So if we do that enough, we can add that data into the model so that it constrains it further, because we're measuring exactly what's going on in the cell. And that's helpful because, again, biology loves redundancy, and so you can capture all of these inoptimal like cycles or other reactions that are going on. And so that's one way to account for the large solution space, is to do the experimental measurement of the fluxes themselves.
Thank you.
Yes, then I don't see any more hands. Then I thank you again for a nice presentation.